Hello, students. So we'll start the chapter crop production and management. In the lecture 3, we will study disease management and plant breeding. So in case of plant, that means whenever we are going to cultivate the plants, at that time, we can see that there will be some insects or there will be some infections you have in the plant. So we have to know how to manage those insects. So we can do one thing. We can use synthetic pesticides. Synthetic pesticides. So, but synthetic pesticides, we have seen that a lot of disadvantage we have. So, what to do now? That means whatever we have to do, that we have to use we have to use some whatever the biodegradable things, and we are not going to use any type of synthetic chemicals as a fertilizer. We can use we can use manures, that is natural manures. So we can use vermicompost, but we are not going to use any synthetic or chemical inorganic chemical fertilizers. Now, plant breeding also we'll study here. So first, disease management. Disease can be a major concern. Just a minute. Disease can be a major concern for organic farmers as it might reduce the crop yields, because always we have the target that yield should be very high. Yield should be very high. The quality of the quality should be high, good quality, and also as whenever we are saying that yield should be high, that means the quantity is large. From this we can say that the quantity is large, the produced quantity is large, and whatever the produced crops, those have a very good quality. Now, if we supply important macro and micronutrients and adopting the crop rotation, that can prevent the plant diseases. Okay, and even the soil is enriched with useful microbes, fungi, bacteria to prevent the harmful organism in check. So we can do these two things to avoid the disease, whatever affect our crop production. Like we can see that there is a tobacco mosaic virus here, cauliflower mosaic virus here, barley yellow dwarf and lettuce mosaic virus, sugarcane mosaic virus, bud blight, maize mosaic virus, peanut stunt virus, leaf curl virus. So there are different types of viruses that can affect our plants. So we have to take care that the macro and micronutrients and the adopting crop rotation we can use. By adopting crop rotation, we can prevent the plant diseases. Okay. So organic fruits and vegetables that are not only the example of successful organic products, recently dairy products that are also organic, and it is very noticeable in our India also now. So they follow strict means of farming, like animals feed only the organic foods. We are not going to give them for better to get better amount of milk. We are not giving them a lot of inorganic things. So we are maintaining one thing, the animals we are feeding with the organic food only. So whatever the animal you are just going to make one farm, that animal should take organic food. Hormones or other genetic engineering practices for high yield are not allowed. We are not giving so much hormones, because these hormones we are using to get huge amount of milk, huge huge amount of milk, or we can use these hormones or some genetic engineering also we are applying, but in this case it is prohibited. In this case they are not allowing us to give the hormones to the cows or give the hormones to the domestic animals, right? So these two things we should think that these two things we should take into account. First is the animals, they should take the organic food only, and next is that the hormones or other genetic engineering practices to get the high yield, we are not going to allow the animals, right? So we are not going to allow the farmers that they should not use the hormones or any other products to get much more milk or much more productivity, right? So this thing has been seen in case of our animal farming also. Now, the advantage of organic farming, that improves the soil fertility. As I have told you, whenever once you are going to grow the crop, at that time you can see that the soil fertility becomes, the soil loses its fertility, and the biological diversity also, it is not maintained. So that's why we are saying that crop rotation is a remedy. Crop rotation is a remedy to maintain the soil fertility. To maintain the soil fertility. Another thing is that the organic farming, this helps us to maintain the soil fertility, and good for this organic farming is good for environment and yields, because the inorganic chemical fertilizer, we are not going to use chemical fertilizer, we are not going to use any synthetic pesticides, so in organic farming, this will help us to protect our environment from the synthetic pesticides. It is not like that whenever you are using synthetic pesticides, it will only affect the plant whenever you are just eating the foods, or you are eating the crops as a food. Then it will affect the human and animal whenever they will consume those type of crops. So this organic farming, that helps us to keep the environment clean, and it helps us to keep the human and animal beings protect the human and animal beings. Okay. Now it prevents the soil erosion. Now here, what is soil erosion? This is actually, it is a form of soil degradation. So the soil erosion, this is actually the soil degradation. And why it happens? This is a natural process, and it is caused by the dynamic activity of erosive agents. And what are the erosive agents? Like maybe some flood, so water is responsible, ice is responsible, snow, that is responsible, air, plants also responsible, animals also responsible, which are grazing on the soil. So these are the responsible things. So that means the organic farming, that will prevent the soil erosion, degradation, and crop failure. So the soil erosion, I have told you, soil erosion means soil degradation, the upper layer of the soil that will degrade, and it is due to the water, it may be ice, snow, air, plants, animals, due to all reasons, it may degrade the upper layer of the soil, okay, the topmost layer of the soil, and it can degrade the soil quality, and it will decrease the pollution, and whole farming can easily rely on renewable energy sources. So here the pollution will be less, and the farming, it can also rely on the renewable energy sources, like it can use only the sunlight. Here you can see that this is a organic farm, and sometimes they are covering this with a sheet, and that sheet will get the radiation from the sun, and it will give the required warm temperature to the particular plants, and it improves the soil fertility, enhances the chemical and physical properties. So we may conclude one thing, that organic farming is the future scope. Now organic farming is going on in India, but this should be done in many fields, and best method for the farmers, which is profitable, also rewarding, and we are encouraging the farmers to do the organic farming. We are encouraging the farmers to do the organic farming, because the organic farming, this is very important, and that will protect our soil from the degradation of the soil nature, and also it will maintain the soil fertility, and it enhances whatever the chemical and physical properties of the soil. Okay, so organic farming we have followed from the ancient time, but we never after that ancient time, because at that time what we have, the requirement is less. The requirement was less because the population was less. But now you can see that the population is so high that the demand for the crops, particular crops are, the demand is so high. That means what we have to do, we have to increase the yield. So to increase the yield, we are going to use so many hormones, we are going to use so many chemicals just to increase the yield. But we are not aware so much that it can affect our human or the animals also, other animals also, who are consuming those crops. So now a little bit awareness comes into picture, and now we are focusing the organic farming. The market for organic foods is growing at a compound annual growth rate of the 20 to 22%. So here we are going to increase the organic farming, and we are encouraging all the farmers to do the organic farming. And here we can see that little bit we have improved our environment also. Now the approaches to improve the crop yield, that the crop yield improvement, here in the crop yield improvement, the plant breeding helps the farmers to select the desired properties of the crops and cross them to produce new varieties. Suppose one crop you have some properties of this crop is good, another crop you have this properties is also good. So we are making the cross, and then whatever the plant we are growing, it has the desired quality. Okay, it has the desired quality and it will give us high yield. So the plant breeding that helps the farmer to select the desired properties of crops. So for these crops, suppose particular one property is good, another crop, another property is good. Suppose this quality is very good, but yield is not good and it has high yield. So what we have to do, we have to cross these two, and then we have to produce a new variety. And this new variety, they will provide the farmers high yield as well as high quality also. So this has made it easier to feed the large population of the country, and there will be no scarcity. So, and also it helps with whatever the new variety or the new property you have incorporated in the new seed. You can see that this is also disease resistant crop. Disease resistant crop. So crop variety improvement, first thing is that breeding. First thing is that breeding. Breeding means if you have the crops, different types of crops, just you take the good qualities of those crops and you just cross them to produce the new varieties. Then you can cultivate them. So you can see that you are getting a large amount of crops with a very good quality. Also, it is disease resistant crop. So that means our approach is good approach, right? Now, the crop variety is improved due to the following reason. First reason is that by improving the crop variety through the crossbreeding and hybridization, the crop yield can be increased. Okay, so here the crop becomes resistant to biotic and abiotic stresses. Already I have told you biotic and abiotic means your bird, insects, all those things. And here, though the crop becomes resistant to biotic and abiotic stresses, for short duration crops, the early maturing varieties can fit into multiple cropping varieties. The new varieties can assimilate to different environmental conditions. So here you can see that the ideal plant architecture, this one, high yield, high quality we require, biotic, abiotic, stress resistant we require, efficient nutrition update also we require. So here crossbreeding we can do, mutation breeding we can do, and transgenic breeding we can do, breeding by genome editing also we can do. So these different types of breeding we can do, where mutation breeding, that means random mutant genesis background is required for transgene free 8 to 10 years. So here we can do mutation breeding, the transgenic breeding, breeding by genome editing, and also crossbreeding. Okay, so breeding is important to get high quality and high yield. High quality and high yield, high yield. This point you should remember, and also disease resistant. This crop we require. So we can do it using the breeding method. Breeding means what? We are taking different crops and good qualities of the crops we are crossing. So we are getting a new type of crop, new type of variety that has so many advantages. Now, what is the plant breeding? So plant breeding, that is the purposeful manipulation of qualities in plants to create new varieties with a set of desired characters. Now, here, this is the definition, and where plant breeding is the purposeful manipulation of qualities in plants to create new varieties with a set of desired characteristics. Suppose you just take one example. You have 10 friends, and if each friend you have one good quality, right? One is very decent, another is very intelligent, another one you have very disciplined. So this type of, that means all the good qualities I am taking, and then I'm just adding it, and we are creating a new person, and that person have all the good qualities of each person. So this plant breeding also like this. The plants with higher qualities are selected by and crossed to obtain the plants with desired quality. And this result in a plant population with improved and desired traits. So here you can see that they have taken a lot of plants. Then the qualities of the, suppose the tallness, suppose the crop quality. So disease resistant, some plant you have disease resistant, some plants you have the quality that is high yield, some plants they require less maturity time, okay, less maturity time or period, less maturity period, I can say less maturity period. Another thing, soil fertility, they are maintaining the soil fertility. And another thing, it may be they can resistant to the climatic climatic variation, climatic variation or adverse, in the adverse climate they can grow. So all these are the good qualities of each plant, and all these good qualities we are incorporating in a particular plant, so in a particular breed. So that is called the plant breeding, right? So that means whatever the new variety, it has disease resistant, it has high yield quality, it requires less maturity time period, it has soil fertility is good, resistant to the climatic variations. So there are so many points you have, and you may consider all those plants, and then you can breed, so that you, whatever the new variety you will get, in that variety you can see that all the good qualities are there. Okay, and this thing is called the plant breeding. This process is called the plant breeding, and this results in a plant population with improved and desired traits. Traits means nothing but, traits means character. Traits means here character. Okay, you can say also the character. So traits means here you can say also the character. That means every good character or every good qualities you are taking from different plants, and you are just growing one new variety, and this new variety is the best one, is the best one where you have all those good qualities, right? Now, plant breeding objectives. Why we should do this thing? The objectives are that higher yield, whatever I am saying, resistant to disease and pests, change in maturity period, elimination of toxic substance, synchronous maturity, photo insensitivity, resistance to abiotic stress, improved quality. So all those things should be incorporated. That's why we are doing the plant breeding. So to increase the crop yield, to raise the plant with desired characteristics like disease, it should be resistant to the disease and plant pests. The change in the maturity period, that is also important. Elimination of the toxic substance, that is also important. Synchronous maturity and photo insensitivity and resistant to the abiotic stress and improvement quality, all those things we require from a plant. And from a particular plant, it is not possible if you are not going for the plant breeding. So that's why the main objective of the plant breeding is that to increase the crop yield, to raise the plants with desired characteristics, to develop a disease-resistant crop, to develop the plants that can tolerate extreme environmental stress. As I have told you, it may be the sunlight, how much you require, or the radiation of the sun, how much you require, it is not consistent. So the consistency I cannot control, but I can control the plant that should be that has some resistance to this climatic environment. So, and types of plant breeding, and these are the different types of plant breeding we will discuss here. And these qualities, before that plant breeding, we have these qualities should be incorporated. Okay. Now, first one is the back crossing. In this, a plant with desired trait is crossed with a plant that does not have the desired traits, but has several other traits. That means one character or one trait you have, and one desired trait, you are taking one plant which has this desired trait, and another plant you have which does not have that trait, desired trait, and then you are making the breeding. So here other traits may be there, so that is called the back crossing. That is called the back crossing. Okay. Now, inbreeding. Here, self-fertilization occurs in the inbreeding. Here you can see that same flower you have and different flowers of same inflorescence we can have. But here we are doing the self-fertilization. And this progeny produced in the same generation after generation. So this helps to preserve the original traits. So inbreeding, back crossing, we have studied till now. So back crossing is what? One plant we are taking which has the desired character or traits. Another plant we are taking which does not have the desired trait but have several other traits also. Then we are making these traits will be crossed, and at that time we can see that whatever the plant will be, the new plant or the new variety will be there, that breed is very good breed. Now, inbreeding technique. In the inbreeding technique, we are using inbreeding technique. In inbreeding technique, we are using self-fertilization. Now, here you can see that the same flower, what we are doing, the progeny that will produce is the same generation after generation, and this helps to preserve the original characters. Here you can see so many breeds are there. So here you can see that this is called inbreeding, and this is the back crossing. This is the back crossing. Now, hybrid breeding. In this, two different breeds are crossed, and to produce the offspring that is more productive than the parents. Here, offspring means the child. Okay. Now, in case of plant also, if you have another plant will be produced from the first plant, that means this is a child. So here, the hybrid breeding, you can see that for F1 hybrid breeding, female germplasm pool here, then female inbreds, then selection on hybrid performances, female parent line, and here also male parent line is coming. After that, whatever we are getting, that offspring or the child, it has some characteristics of the female and also the characteristics of the male, but which one is dominant, that will be transferred. And then is the mutation breeding. Then we can study mutation breeding. In this mutation breeding, the mutation is in plant genes result new variety. Mutation can also be induced in plants by exposing them to chemicals and radiation. So in case of mutation breeding, what we are doing? Mutation breeding, what we are doing? Like mutation breeding, sometimes it is also called the variation breeding. The mutation breeding, or sometimes we are calling it variation breeding. And it is the process of exposing the seeds to chemicals and radiation or enzymes in order to generate the mutants with desirable traits to be bred with other cultivars. So here, that means what we are doing, we are exposing the seeds we are taking, then we are exposing it to the chemicals, radiation, or enzymes. Okay, and here to generate the mutants with desirable traits. And plants created using this mutagenesis are sometimes called mutagenetic plant or mutagenetic seed. Mutagenetic plant or mutagenetic seeds. So here the plant creating the mutagenesis are called mutagenetic plants. And here you can see that there are, that means the seeds, they will be exposed to the chemicals or radiation and enzymes, and we are the mutants, we generate the mutants with desirable traits. So we will get new type of mutation breed. Okay, so this is the mutation breed. Now the genetic engineering, it helps us to produce the crops with desirable traits by inserting the gene of interest within the crop DNA. Now here you can see in this picture what we are doing. Suppose this is, now here the antibiotic resistant gene is this one. Okay, chemosine. So this is the antibiotic resistant gene, and this chromosome is here. So whatever we are doing from this, we are making some plant cell, then we are transform the cell to the, then culturing, tissue culturing we are doing here. So we are culturing these cells, then we are able to grow the plantlet, then we are going to grow the transgenic plant. So genetic engineering that helps us to produce the crops with desirable traits or the characters by inserting the gene of interest within the crop DNA. So such crops are known as genetically modified crops, and this is actually BT crops. We can have BT brinjal, we can have BT corn, we can have different types of BT crops here. So genetic engineering we are using for this. Next, plant tissue culture. This is also very important. Whenever we are going to produce new variety, the plant tissue culture will help us. The plant tissue culture is a new addition to the methods of plant breeding, and it is developed around 1950s. Plant tissue culture. And this plant tissue culture, here whatever we are doing with a knife, we are just peeling off this portion. Then this tissue we are taking into a solution, This tissue sample that will placed in the agar growth medium containing nutrients and auxins. Then you can see that samples develop tiny plantlets. And after that, if you just these plantlets you take and you just take one container where you have the soil and you just planted all those into the compost, you can see that you can have a large number of very good plant. So this is called the tissue culture. This is the method of tissue culture. So first what we are taking, this is the main mother plant. Now from this tissue will be collected. Now tissue sample scrapped from the parent plant. Now this will be transferred to the this sample will be transferred to a container where you have a medium containing the nutrients and auxins. Then we are you can see that the samples develop into tiny plantlets. Then we are taking out the plantlets and we are just adding some compost here, and then these plantlets will be implanted. So you can see that a huge number of plants you can have from this, right? And this thing we are doing in case of our different farms also, right? Now this tissue culture, this makes the parts of the plant to generate multiple copies of the plant in very short duration. It will take very short duration. This is one advantage. Very short duration it will take. And another thing is that similar plants, whatever the plantlets you have here, they are all similar. That means all similar traits uh, has been there. All similar traits are there for each plant. The technique exploits the property of totipotency of the plant cell, which means that any cell from any part of the plant can be used to generate whole new plant. So here, this is the portion of the plant which we have used to make a huge number of plants. Okay. Now the types of plant tissue culture. Seed culture, embryo culture, cellus culture, organ culture, protoplast culture, anther culture. Now here you have, these are the types of plant tissue culture: seed culture, meristem culture, protoplast culture, organ culture, pollen culture, callus culture, cell culture, embryo culture. Okay, so these are the different types. Now procedure already I have described. You just peel off this tissue, take it into the solution where you have nutrients and auxins. Then you have to transfer it to the samples. Then you have to transfer it. After certain time you can see that it will grow like this. After that you transfer it in a compost, so you can see similar plants will be there. So the X plants are cultured in vitro on a nutrient medium that caters to fulfill the nutritional requirement. So this is the procedure of plant tissue culture. Now the nutrients medium must provide you. That means we are taking some nutrient medium. So the nutrient medium that must have macronutrients, also micronutrients. Macronutrients means it includes the elements like nitrogen, phosphorus, potassium, calcium, sulfur, and these are required for the growth and morphogenesis. Okay, so macronutrients we are requiring, and the macronutrients are what? Here I am writing the macronutrients. Macronutrients, please remember the macronutrients. We have nitrogen that we have to provide, phosphorus and potassium, calcium, sulfur, These are the macronutrients, and micronutrients, this we require iron, we require manganese, we require zinc. These are the micronutrients we require. So these macronutrients, this will help the plant to growth, for the growth and morphogenesis. And these micronutrients, they are crucial to the growth of tissues, growth of the tissues. Okay, so here it has been seen that if we are just taking one cell here, then we are putting it in the nutrient media, then it will be created, then embryo will be created. This embryo, from this embryo, the plantlet, and you can see that the similar plant you can get. Okay. Now carbon or energy source. So this is the one of the most crucial ingredient of the nutrient. Now sucrose is the most widely used carbon source among the carbohydrate and serves the carbon, hydrogen and oxygen. Vitamins, amino acid and other organic salts also required. So nitrogen, calcium, cytokinin, oxygen, these are also required when in the solution. Okay, so these are also required in this solution. Now apart from this, the culture media also serve as a medium for supplying the phytohormones or plant growth regulators. These are the plant growth regulators to the issues which bring about their morphogenesis as per requirement. And the tissue of the plants first lose their specificity to form a hard brown lump known as callus. So here you can see these lumps. Okay, here you can see this. Okay. Now this will split to develop a plant organ, then hold a new plant depending upon the quality and composition of photohormones supplied. Then we are going to supply the photohormones. Here you can see that bud, stem, root you have. From this leaf you are taking, this is the cell structure, and from this cell you have taken some tissue sample. Then we are forming the callus. Then this callus separated and single cell cultured. Then you are further culturing regenerated new plantlet. So the entire process requires strict aseptic condition, and that we should maintain. Aseptic condition that we should maintain, right? So in this case, whatever we are seeing that this, in this case we require this leaf tissue, and then we are forming this callus, then it will be separated, and then it will be, you can see that some plantlets will be there. Now aseptic means the absence of the germs, because if there will be germ, then this will just destroy whatever the cells you have collected, that will just destroy it. So you should keep in your mind that this aseptic condition should be maintained. This aseptic condition is very important in this case. There should be no bacteria, virus, microorganisms that can cause the diseases. It should not be there, because they can ruin the entire batch of plants. So be careful about this thing. Plant tissue culture is a very good type of method, but here aseptic, we should maintain the aseptic conditions. Now the use of plant tissue culture. Tissue culture is used to develop thousands of genetically identical plants. Okay, so plant tissue culture we are doing for higher yield, independent of environment season. The seeds should be independent of environment season, the plants, and potential for low cost production, harvest in short duration, production of novel compounds, and round the year availability, continuous supply of products with uniform quality, Also conservation of natural population and sustainability. So this method offers the plant tissue culture. It offers several advantages, and it can be used to develop disease-free plants and disease-wrought plants from the disease-wrought plants by using their meristrates or as the explants. So these are very important method. This is the very important method which gives us higher yield, which gives us those plants are independent of the environment season, and this potential, it has potential of low cost production. It has harvest in short duration, that means maturity time period is less, and production in the novel compounds and round the year availability can be there because it is resistant to the weather also. So we can avail it throughout the year, and continuous supply of the products also we can get. Okay, so these are the uses of plant tissue culture. With that, now I'm ending my lecture. Next time we'll continue this crop production and management chapter. Till now, till then, bye bye. Now, thank you.